Hi, everyone. So today's lecture is going to be recorded because I'm not feeling super well, and how appropriate for us to be talking about disinfecting and sterilizing our environment. So hopefully none of y'all get sick as well. So we talked previously about the differences between disinfecting and antisepsis. And the difference there was that antisepsis was to be used on skin versus disinfecting would be used on non-living surfaces. But there is still more to differentiate between disinfecting and what we refer to as sterilizing or sterilization. And we'll talk about the reasons why we would use both of those techniques in today's lecture. But just for starters, in the real world, it's important for us to use disinfecting and or sterilization for things like food preparation. We talked all about Typhoid Mary and how she would contaminate her food and then pass that on to the people who she worked for. But we've also talked a little bit about the importance of disinfecting and sterilization in healthcare settings. For instance, if there is someone who has an open wound and requires surgery, we want to make sure that they don't get infected from some sort of contaminant in their area. But we've also worked with both of these ideas in the lab as well. So for one, disinfecting, we always spray down our lab benches before we start and after we are finished with any of our experiments so we can prevent contamination. And when I am preparing for you all to come into lab, I utilize sterilization techniques in order to prepare some of the media or the culture broth or culture plates that we work with in the lab every day. In addition, whenever we are done with the reagents, like all of those plates that we are throwing out or the tubes that we are no longer using, I need to sterilize all of that biohazardous waste through use of an autoclave. And so we'll talk about that today as well. And so just again, the differences between controlling microbial growth in terms of antiseptics versus disinfectants. It really comes down to the type of surface that we are looking to treat. For example, an antiseptic is probably going to be a lot more gentle than would be a disinfectant because we obviously want to protect little babies over here versus if we are just trying to treat a sterile stainless steel surface. And even further, a disinfectant is probably going to be more gentle than a sterilization technique. And so that's really what we will differentiate today. But ultimately, all of these techniques, whether it be antisepsis, sterilization, or disinfection, it really comes down to preventing disease transmission or microbial spreading. And there are a number of different ways that people can be infected, obviously through direct contact, through shaking hands or kissing. We all know that during our COVID era, we all wore masks because COVID was spread by the air and droplets through sneezing or coughing. And today, we are going to focus on indirect contact through what we refer to as fomites and by food, which we can refer to as a vehicle. And we will talk more about those terms on the next slide. Now, eventually in class, we will also talk about vector transmission. So that is like through insects or through spreading diseases through like things like rabid animals or animal bites. So that'll come later on in the course. And so to define these two words that we just mentioned, a vehicle versus a fomite. A fomite is going to be an inanimate object that when contaminated with infectious agents can transmit disease to a new host. So this is really anything in your immediate surroundings that potentially could hang on to bacteria or viruses. And if someone else comes around, touches that surface, and then touches any type of their mucosal membranes or eats some food, they could then transmit that disease into themselves. So, perfect example would be like our benchtop tables in the lab. A lot of you swabbed phones or toilets or handrails. So, all of those examples that we swabbed in the lab would be considered fomites. A vehicle, on the other hand, is a broader term. So, this is any contaminated inanimate object or non living object or resource that can transmit disease. So, this opens up the realm beyond just fomites, but also things like water and food. And so, Typhoid Mary was transmitting typhoid fever to all of her, the people who she worked for through a vehicle, through her food. And so now let's also differentiate the difference between disinfection and sterilization. So we talked about antisepsis versus disinfecting in terms of the surface, but when we're talking about disinfection versus sterilization, we're not talking about the surface so much as we are talking about how clean the surface will be once we are done. For example, in lab, anytime we spray down our surfaces with that Lysol spray, we are disinfecting that surface. So that's removing the majority of microorganism contaminants on that surface. It's not necessarily completely obliterating any type of microbial presence, and even on all of the hand sanitizers or Lysol sprays or any type of other disinfectant that you would get at the stores, they never claim to be able to clean a surface 100%. A lot of times they'll say it's 99.9% effective. But this is just to say that we are not going to be able to completely sterilize a surface through means of disinfecting. Now, that is in contrast to proper sterilization techniques. And this would, in fact, kill all microorganisms on or within a surface. So, an example that we can think of for sterilization would be, for instance, the canning process. When we take, whether it be canned corn or green beans, you'll notice that the shelf life on those items is really, really long. That's because we can expose those cans to so much heat that it does, in fact, actually guarantee we are killing all microorganisms within that can, thus properly and effectively sterilizing that surface. So for starters, let's talk about some common disinfection techniques. And what we'll notice today is that there actually are going to be some overlaps between disinfection and sterilization, but the difference really comes down to the concentration of the product or the length of time that an object is exposed to such a technique. So disinfection, the two most common ways that we approach this is through either chemical exposure or some sort of physical exposure. So for chemicals, we think about a lot of things that are common household items like bleach. I'm sure most of you have some sort of bleach to help clean your bathrooms. In the lab, we use Lysol spray, but in a lot of other labs, we actually can use ethanol as well. It works in a very similar way. So if you're looking at this, you're probably thinking, ethanol was also an example of an antiseptic, and you'd be absolutely correct. Some of these products can actually be used for both, and again, it comes down to the concentration and the amount that we are using. For instance, a rubbing alcohol used for antisepsis is going to be at a lower concentration than would be an ethanol that we are going to be cleaning our benchtop with. Two other examples using physical means include heat, like if we were boiling something, or UV irradiation. And we'll talk a little bit about that today and why it's actually maybe not used so widely today. So, one of the most common things that probably all of us do, at least at this point, I hope we all know how to, would be cooking. And disinfecting food and disinfecting these vehicles are going to be very important for keeping us healthy. For example, you have probably all heard that chicken needs to be cooked to 165 degrees Fahrenheit, or beef needs to be cooked to 160 degrees Fahrenheit. And have you ever stopped to wonder why are those specific heat recommendations mandated? As it turns out, it actually doesn't really have anything to do with the material that we are cooking. In terms of chicken doesn't taste better if it's hotter than if it were beef. And beef doesn't necessarily need to be cooked to 160 degrees because otherwise it's inedible, but rather we cook and we recommend cooking those meat products to those specific temperatures because of the microorganisms that commonly live on those surfaces. For example, we cook beef to 160 degrees because that is the temperature required to kill off most E. coli bacteria. And so, because E. coli commonly lives on beef, we want to make sure that we eliminate the E. coli. We don't need to cook it that extra 5 degrees because at 160, that would be sufficient. Whereas with chicken, we've probably all heard about salmonella, and that is best killed at 165 degrees Fahrenheit. And so when we are cooking and having these safe recommendations for meat temperatures, it comes down to the types of bacteria that live on those surfaces, not so much the meat itself. And when we're thinking about storing meat or keeping things safe for longer periods of time, anything above this 60 degrees Celsius is going to keep bacteria from growing. Or if we are trying to store food, keeping it below 5 degrees Celsius. The reason why is because if we think about the microclimate of bacteria, and I'll just draw a little graph right here, and let's say we have temperature on our x-axis and maybe growth rate on our y-axis, we oftentimes see a little bit of a bell curve like this, where these middle are like meso temperatures is where the bacteria grow the most. And so if we keep things really hot, so in this upper quartile range, or cooler, we can actually limit that bacterial growth. And so keeping food either hot or cold is going to be the best way to keep food from growing microorganisms. But there are some instances where food needs to be prepared and disinfected before it even gets to your kitchen table. An example would be like fresh cow's milk, or things like honey or cheeses, any type of dairy product. We want to make sure that it's not harboring microorganisms before it gets to our table. But if we're thinking about heating meats in order to disinfect that from our fridge to our kitchen table, we can't really do that for all food items, and milk being the perfect example of that. If you have ever tried to heat milk on a stovetop, whether you're trying to make hot chocolate or some sort of roux for a soup or a mac and cheese, perhaps, you'll notice that if the milk gets hot enough, the proteins actually change in that milk. It starts to curdle, the taste actually ends up changing, and we don't consider that to be really what we want for milk, for instance. And the same thing with things like cheese. If we heat cheese to a really, really high temperature for long, it's going to change its properties. Like if you've ever made a grilled cheese sandwich where all that cheese is nice and melted, and then you go to reheat that sandwich, the cheese kind of changes its texture. So, all of that to be said, we can't heat certain things for this higher temperature. And so, we have developed pasteurization, which is the process by which we can heat all of these items, but for a shorter amount of time to still be effective in killing off microorganisms, but not actually changing the innate properties of the food product. And so we can see here, this is just sort of a cartoonish example of what this pasteurization process looks like. But we can imagine that we get fresh milk going into our pasteurization process over here, and it goes through a series of coils, first of which is undergoing heating. And this heating process is very quick. Like in cases of milk, it's only going to be for about 15 seconds. Immediately afterwards, the milk is then cooled to 4 degrees, which is about the temperature of our fridge at home. This process of very quick heating and then very rapid cooling actually allows for the microorganisms to be killed off, at least for the most part. Now remember, disinfection is not necessarily going to be sterilization or sterilizing our milk, but we are going to reduce those microorganisms so that it'll be safe in transport and safe in our fridge, at least by what we would expect that best by date to be. Eventually, that milk is then filtered out and put into jugs or cartons for us to purchase at the store. Now, when we are thinking about non-liquid products, that actually undergoes a different type of disinfection process before it gets to the grocery store. Like, think for instance, we want to make some tacos at home, and so we pick up a packet of ground beef, and we look at the ground beef in the store, and it's already been cooked. Honestly, that doesn't sound very good to me. We want to be able to cook our meats to how we prefer it, and so that it's fresh for when we make our tacos. For the same reason, stores and processing plants can't use super high heat in order to prepare their meats because that's going to cook the meats and again change that composition to where it's maybe not going to last for quite as long or it's not going to taste quite as good for us once we finally purchase it. And so, because meat isn't necessarily a liquid that we can put through a pasteurization machine, instead we use something called irradiation. This process of disinfection uses UV light in order to destroy DNA. And so, if we're thinking about UV light, that's just like the sun outside, and why it's so important for us to put on sunscreen as protection from that UV light. Ultimately, we know that long-term sun exposure or really bad sunburns can lead to an increased development of skin cancers. And skin cancer is really a lot of times from mutations in our DNA. Now, if we do this or use this process intentionally, we can purposefully kill and destroy the DNA of an item, whether that be the beef DNA or, as it turns out, the microorganisms that are on that beef. By putting it through this process of destroying DNA, we would thus be able to effectively kill microorganisms. Now, again, this is a disinfection process, so it's possible that we're not going to be able to kill all of the microorganisms on the surface. Now, ultimately, if we're thinking about ground beef, all of that meat is going to be mixed around and it's going to be kind of mashed up. And so there probably will be some microorganisms within the ground beef that have withstanded this UV light irradiation process, which is why people always recommend that if you are cooking with any type of ground meat process, that you cook it for a longer amount of time so that it's cooked all the way through, because there is a higher likelihood of microorganisms to be within that ground beef versus if you were to get a steak that is just going to have microorganisms on the surface. Now, we've talked more about the vehicles in terms of food and resources, but there is disinfection of fomites, where we would just use something like a Lysol spray or a bleach or a ethanol diluted spray. And that is demonstrated here. So disinfecting can be any one of these strategies that we've mentioned. So now this brings us to sterilization methods. Sterilization is going to be the complete removal of microorganisms on a given surface. And you'll notice here that a lot of these things are actually the same as we've used for antisepsis, like for instance, this quaternary ammonium salts, or disinfection, disinfectants, like it's even mentioned right here, concentrated disinfectants can actually work for sterilization. In addition, we have things like heat and UV irradiation, which we've also mentioned for disinfection as well. So, in addition to disinfecting things like meats and cheeses, we can also sterilize food. And one of the most common methods is the canning process. There are a few different types of methods, especially if you've ever attempted at-home canning. I personally haven't, my sister has. But we can take jars and subject them to high heat. Now, a lot of times this is some sort of steam heat or used with water. So we have this boiling water method here, and that's actually demonstrated right here in this image. And we also have a pressure cooking method, where it's essentially not sitting in a bath of water, but we are using extreme pressure, extreme heat, in order to eliminate microorganisms within a jar or a can. In general, this canning process includes a temperature of at least 121 degrees Celsius, 14 psi, so that's pressure, and then for at least 15 to 20 minutes. And so this is going to be slightly different from our pasteurization process, which again is only going to be for a matter of a few seconds in terms of the heat. And it turns out in lab, we have actually worked with sterilization as well. So when we are using our Bunsen burner in order to sterilize our microbiological loops, we use flame heat in order to incinerate basically anything that could be existing on that loop. Same thing with our hockey sticks that we used for spread plating, where we have the ethanol at a 70% concentration. We then pass that through the Bunsen burner. So a combination of a chemical sterilization plus heat in order to completely eliminate any type of microorganism. There are two other methods for laboratory sterilization. The one being dry heat, so that's not going to be an open flame, but rather essentially just an oven. So we use an oven in order to sterilize any type of metal or glass, anything that is you can think of as oven safe. And we use that at about 150 to 170 degrees Celsius commonly, which ultimately equates to right around 300, 350 degrees Fahrenheit. So just like your oven at home. Now, the one thing that we haven't talked about so much just yet, but we have a steam heat autoclave. And so essentially, we can think of this at home essentially being like an Instant Pot, where it's just a pressure cooker. We are going to be adding heat, pressure, and time in order to sterilize equipment. Now, this steam heat autoclave is going to be at a lower temperature than a dry heat oven or what we subject our microbiological loops through the flame heat. But with that added pressure and extended period of time, that can contribute to proper sterilization. So before you get any of your plates, I make up some sort of broth or agar or media in these glass bottles. I put them right here inside of this autoclave shown on the lower right. And I end up letting that sit for about an hour and a half, and then it's sterile. So then when I pour the individual plastic plates for you, that agar that we have isn't already going to be growing things by the time it gets to your benchtop. Now we also have laboratory sterilization through UV irradiation. So what you probably remember is we talked about irradiation in terms of disinfecting things like ground beef or other types of meats. So this UV sterilization is no different than what I've explained before. It utilizes UV light in order to basically annihilate any type of DNA that's present on surfaces. Now, the difference here is that it's going to be for a longer period of time, and the objects that we are hoping to sterilize aren't going to be as deep or dense or complex as something like a pound of ground beef. You'll see here in this picture, although I'm realizing it's quite blurry, it is just a stainless steel laboratory bench surface. Maybe we have some equipment over here like a tip box or a waste beaker, but that light can penetrate through those surfaces. Now, we don't actually use UV light sterilization anymore, not because it doesn't work, but because the NIH and the CDC have determined that it actually poses a lot of risk and harm to scientists as well. You can imagine that this UV light, it's going to be in our laboratory space. And unless that sash, this like glass window, is pulled all the way down safely, that UV light could potentially, we could expose ourselves to it, and we could end up suffering some consequences of that extended UV irradiation as well, thus leading to potential cancer development, which obviously we don't want. So although it's an effective means of sterilization, really it's just recommended to utilize things like bleach or other type of ethanol cleaner on your lab surfaces to do basically the same thing, but is safer for us. And last but not least, there is one other technique commonly used in the laboratory setting, and this is filtration, which really is pretty self-explanatory. If we have a filter, so think for instance, even a colander, when you go to drain pasta noodles, you are going to keep all of your pasta, at least hopefully, in the colander, and that liquid is going to pass through. Same thing can be used for media or other types of liquids used in the lab. If we filter those liquids and we use a filter that is small enough, so right here we have 0.45 microns. Now, if we remember, this sort of size limitation for a microorganisms is anything below 100 microns. This is going to be a pretty effective filter. So it might not filter out all the virus particles, and actually that's how viruses were discovered. It might not filter out all of the very, very, very small bacterial cells. However, we can imagine that for at least common exposures and sort of common presence in a laboratory setting, a filter of this size is actually going to be able to filter out all of the microorganisms. And so the microorganisms would get stuck right here to this filter, and the liquid broth, media, water, anything that we're trying to filter out is going to end up in the container on the bottom, sterile and ready for use. So as always, I have included some study questions on the slide. I will work to get a written transcript ready for those of you who maybe don't learn quite as well by watching videos, but I hope that this has helped. I hope that you can pause it or play it as much as you need to get all of the information. But in terms of lectures that were good to watch over video, this is probably one of the best ones because it's pretty straightforward. But as always, if you do have any questions, please don't hesitate to come into my office. I'd be happy to answer any of them that you have. Have a great day.